you can wait and join the study group if you haven't done so. And after the lecture, I'll be available for that, for the study group. Okay? Now, today's lecture is in two parts. The first one has to do with artificial nanotubes that you create using the optical tweezers, attaching a microsphere that is trapped by the tweezers to a cell membrane and pulling it. And that extracts what is called a tether. And you do this, and you can measure the force of the optical tweezer, which I explained yesterday, and the equilibrium radius of the tether, both at equilibrium. Now, a theory which I'll develop now, called Helfrich-Cunningham theory, then gives both the surface tension of the membrane and its bending modulus, kappa, directly from the values of R0 and F0 by these two formulas. We'll discuss also how the tether grows and how its shape evolves as you pull. And also, it depends on how the cell microsphere contact is established. The theory here assumes a point contact. But in real life, it's not a point, but it's a little surface, and one has to take into account the effect of that. Now, why is it so important to measure sigma and kappa? Because they are related to cell function. For instance, we are now studying how these constants evolve for the case for following not a fully developed cell, but a growing cell and how it evolves during the cell growth. Okay, the second part has to deal with spontaneously emitted nanotubes. The cell produces them spontaneously. You don't pull on anything. And those I call more generally cell membrane nanotubes, CMN. And so the second part is about their structure and their functions. And we'll see that they play an important role in infections by essentially all pathogens. They also play an important role in cancer. And we'll study them in particular for neurons. So that's today's program. Okay, so yesterday I introduced the elastic constants, and this discussion is based on the paper by Dereni, which is quoted there, an excellent paper. So now consider a tether which was extracted with a length L, a radius R, and the pulling force F of the optical tweezers, but the values at equilibrium, which are with index 0. So under these conditions, the free energy must be a minimum. So to determine the equilibrium, we must minimize the free energy subject to the experimental constraints, which are introduced as Lagrange multipliers. The total free energy is the sum of three contributions. First, the free energy associated with bending, which is the surface integral of half the curvature squared. Sorry, this is a kind of generalization of Hooke's law, where you have the bending the torque and the bending modulus in the analog of Hooke's law. So that integral must be extended over the whole area of the bent membrane. Okay. And 2h is just the curvature. And the area, if you have a cylinder, is 2 pi r L. Now, the other term is the free energy associated with increasing the area of the membrane, which resists by surface tension. So you must do work against that. And so that's given by sigma times the area. Sigma is the surface tension. And finally, as you're doing this, you have work, which is performed by the optical tweezers to pull a tether of length L, which is minus the product of F and L. So you add all these three contributions, and you get the total free energy that must be minimized, which is, the first term is that Helfrich-Kanem term, the second term is the surface tension, and the third term is the work. And so the Helfrich-Kanem contribution, together with the surface tension, is the bending modulus divided by twice the square of the radius, plus the surface tension, total area of the cylinder, 2 pi RL minus FL. So you just minimize this free energy, differentiate with respect to L, solve, and you get exactly the radius R. 0 is square root of kappa over 2 sigma, and F0 is 2 pi kappa over R naught. And therefore, you solve with respect to sigma and kappa, and you find precisely these two formulas. Sigma is F naught over, oh, here is written incorrectly. It's actually 4 pi R naught, and kappa is F naught R naught divided by 2 pi. So by measuring these equilibrium values, you can determine the membrane elastic constants. There are two assumptions here. One is the tether is just an empty cylindrical membrane tube with no cytoskeleton inside. That is not quite correct, but it's not yet determined the effect of having actin and sometimes tubulin inside the membrane. And the other assumption is the initial contact between the microsphere and the cell is just a point. And that's not realistic either. Okay, but before discussing that, we can ask, how does the tether grow as you are pulling? Okay. Now, the cell membrane, as a rough approximation, may be thought of like a soap bubble, okay? And we all know the experiment in which you have a soap solution and you drop a ring in it and pull, and you know what happens. It grows outside something analogous to that. And what grows is, by minimization of just surface tension, is a minimum surface of revolution, which is a catonoidal shape. So if you continue pulling more and more, this would tend to an infinitely thin wire. However, that's impossible because that would give an infinite, remember that the radius appears in the denominator in the other term, so that would give an infinite contribution. So that the shape evolution finally, as a cylinder, is a compromise between these two, between surface tension and bending radius. Now, you can compute the shape evolution, and you find something like what is shown in this figure. This is, let me go a little bit lower. If you follow this here, this is the ratio of the R over the final equilibrium value, and this is how much length Z you have pulled. So as you are pulling, it evolves, and it begins like a sort of catenoidal shape, but finally, at some point, this makes a kind of bifurcation to a cylindrical tube. And that's the final shape for the tether. Okay. Now, if you compute it this way, you find that this kind of phase transition takes place as a kind of, with a kind of overshoot, because eventually there is a plateau, okay? And why is there a plateau? The plateau means that you keep pulling, but the force is increasing and the length, the force remains constant, but you keep pulling, and this is the plateau. Why is that? It's an elongation at constant force because the cell membrane has a reservoir of extra membrane in various ways, okay? There are things like, okay, the membrane, instead of going straight, here is the inside, and therefore here you have extra membrane available. So that's what is happening there. Okay, if you compute the overshoot, it's given by the overshoot force divided by the equilibrium force is 1 plus 1/2 of the radius of, oh yeah, I'm sorry I didn't mention this. This is the fact that it's not a point attachment, but the microsphere is attached to a whole area in the cell membrane. And that area has radius RP, P for patch. Okay, so this is what you compute in this way. Now, the next thing is you measure this and find it, results by experiment. Here are results from an experiment that we did at our optical tweezers lab. This is the reference. And you see that it keeps rising, the force, but instead of having this slight 13% overshoot, there is a complete, very sharp drop to the plateau. The plateau is the effect, and so here you are at the equilibrium force. Now, why does this happen? Why is the drop so sharp? And that has to do with precisely the fact that it's not a point attachment, but it's an attachment by a patch, a kind of circular patch of radius R sub P, P for patch. Now, you can estimate what this is if you remember that since the surface tension is sigma and the attachment has a radius RP, the maximum force that the membrane can produce, can support, is 2 pi rp times sigma. Then you use the fact that sigma is given by the Helfrich-Kahnum formula, and you find that the radius, the ratio of the maximum force to the equilibrium force is just the radius of the patch divided by twice the equilibrium radius. And usually, the patch radius is much larger than R0, and that's why you get this very sharp drop. Okay? That's the explanation. All right. Okay. Now, I come to the second part, which is the spontaneous nanotubes. And those I call CMN. The first time they were detected in the laboratory was in a paper by Rustam and collaborators published in 2004. They were using PC12 cells, and they reported that they saw, and this will show below, that distant cells were connected by straight connections, creating a complex network among them. They named these nanotubes tunneling nanotubes, TNT. That has nothing to do with the explosive. Okay. Now, how do they get formed? It's very interesting, and it's shown in this image. Here is the time in minutes, and you will see how to form a nanotube between this cell and the cell above it. Okay, so what is happening is that the cell here that is trying to get attached extends these kind of things like philopodia, which grow in different directions and move as if they were probing the environment. They're trying to find the other cell, and here the time is shown. After 3 minutes, this philopodium-like extension gets in touch with the other cell, and as soon as that happens, you form the TNT. And you see that the image of the TNT here is very, very tenuous. Hardly you can see it. I'll comment on this in a moment. Okay. Now, well, in fact, I can explain this immediately. You see this in a microscope, although the thickness of that tube is well below the resolution of an optical microscope. So what you're seeing is, you're seeing it by scattering, if you like, by diffraction. Okay? Now, Rustam and others observed several features of these TNT. First, they found them to be straight, as you can see in this arrow. The length were up to several cell diameters, so it could be several microns. And the radii, they estimated between 25 and 200 nanometers. And as I just mentioned, this is below the optical limit of resolution of the microscope, so it's just seen by diffraction. And they determined that inside these nanotubes, there is filamentary actin Only that, no microtubules. Second, they are not like Philopodia. Philopodia are going along the substrate, but these are above the substrate, okay? And the other observation is that the cells that are connected, the TNT penetrates within the cells on both sides. So it's linking the cytoplasms of the connected cells. Okay? Is that clear? All right. Now, in our lab, I show you our first observation of these TNTs. And what we used was a brain tumor, which produces glioblastoma cells. That they were cultured, that it's U87Mg. We placed them, when you put them, you see all this has to be done within a recipient with fluid. So you first, as they drop, as you first put them inside, they're just spherical. Now, you wait for 24 hours, and this is what we saw. Okay? You can see that these cells are connected by several TNTs, and this distance scale here is 10 micrometers. And we were able to measure the radius, okay? So we thought that it's typical of the range that we had. Okay. Now, in a paper after this, and the reference is given there at our lab, we studied the structure and the elastic properties of these TNTs. Okay, here is the average TNT radius that we measured by electron microscopy. It's 48 nanometers, so it's just within the range that Rustam and the others mentioned. Now, we were doing this very early after the lab was installed, and we didn't have much experience. So we had to keep the cells, any good cell biologist knows, under optimum conditions, and therefore, in particular, you must have the right percentage of CO2. Well, we didn't have that good enough. So what happened was that one of the cells was undergoing apoptosis, which is cell suicide. And you can see this in the image. Well, in the video it shows much better, but you can see that it's sort of shaking in all directions, okay? That's typical of an apoptotic cell. Now, what's most important in this picture, and we have the video, but I didn't get it, is that it is connected to another cell which is below, it's not seen here, by a TNT. And you see here, okay, that there is a, how you call this, I forgot the name now, but it's what usually encapsulates something, and it travels from the cell below to the other cell. Okay? So this is observed only under stress, when there was no CO2 available. So the guess is that the cell here is sending a distress signal calling for an apoptosis signal from the other cell. Okay? Good. Now, what we did after that, we had that TNT extended, and we asked, what happens if you now use the optical tweezers to pull not on the cell, but on the TNT itself? Okay? All right. We had a surprise here. It appears, but it's not easy to see. Perhaps if the lights are off, you can try to see it better. But maybe I can try to show how it is. Here is the microsphere, and we're pulling on the TNT itself. And you can see that as you pull, it sort of gets in a V-shaped form, okay? As if you were having something dropping. And then suddenly, instead of continuing to extend this, it breaks into two. Is that visible? I hope so. So we call this a V2y bifurcation. Okay. Now, it gives the image here is the force as a function of the distance that you're pulling below. And you see that initially it grows, but just at this point, there is a break, and that's precisely when the V2y bifurcation occurs. Okay. This will be important later. Okay. Now, I'm sort of directly going to the other part, because now I'll show that these TNTs and CMNs are exploited by essentially all known pathogens to propagate infection from one cell to another. Okay? So, first example. Okay. This is with bacteria, and the TNT, the word that I wanted to mention is a vesicle. What you saw before was a vesicle propagating. Okay, now here is the scale, and there is a TNT which is connecting the bacteria, and you can see a kind of network here. And first, it leads to exchange of constituents of the cytoplasm. Second, they showed that it allows transfer, non-hereditary, of antibiotic resistance between the cells. Third, amazingly, they are formed between different species, for instance, Bacillus subtilis and Staphylococcus aureus. And even more, they are formed between Bacillus, E. coli and Staphylococcus aureus. And they may also play a role in collective formation with this connected network of a biofilm. Here are the references. Okay. The next demonstration of how they allow the propagation of infection is here. And that's now is with HIV. And in the paper, which is references here, the HIV spreads along between T cells along essentially the tunneling nanotubes. I think it's very difficult to see, but there is a TNT connecting these cells, and you can follow how this is done. The GAG protein in HIV-1 gets fused with this is shown with the green fluorescent protein. It moves along the nanotube from the infected to, which are the red ones, to the uninfected, which are green here, Jurkat T cells. And they showed that this increases the infectivity by factors of over 100, okay, the magnitude of the infectivity. Okay, now the other example with the other kind of pathogen has to do with prions. And you know that prions are misfolded proteins that produce diseases like the mad cow disease. Here is the path with the mad cow disease. They travel from the digestive system where the contaminated meat was introduced, and they travel all the way to the brain. And one can also follow, I didn't put here the video, but here is the prion. You can see how it travels all the way to a brain cell, and it penetrates within. You can also see it in the video. Okay? The next very interesting paper was to show, because all of these essentially were done in the laboratory, but they were shown to exist in vivo. In fact, the first place where this appeared were in the probably earliest CMNs that were observed, which are very, very thin, which were called at the time cytonemes, and this was in Drosophila. But in this paper, the reference is here, they were seen in vivo. Where and how were they seen? Well, this was an inflamed cornea, and the cornea is transparent, so you could see through it inside, and that's why you can see it in vivo. So the inflamed dendritic cells are connected by TNTs and arrows here. Here's one TNT, here's another one. And as you increase the antigen, say the bacterial toxin, you increase the number of TNTs. So the conjecture is that they may serve to transfer antigen receptor complexes between widely separated dendritic cells, which are the cells that appear here. Okay. Now, I'm going to talk about a connection with neurons. So first, some pictures of the structure of neurons. As you know, they have a cell body called the soma, and then they extend dendrites, which have several lateral branches called spines. And the other thing is that they connect to other nerve cells or to muscle through a very long extension, which is called an axon. And here are nerve terminals, for instance, called boutons. You see all this sort of in a magnified drawing. You see here is the cell body with the nucleus and, well, several internal structures. And you can see the extension here of the dendrites. Here they're connecting two different nerve cells. And here is the beginning of the axon, which would go well below here. And another image, or rather a picture representation, is here, where you see a very important feature, how the connection takes place, okay? And, well, here it is, this connection is between the axon and the receptor in the other cell. They get very close together, but not quite touching. This is called a synapse or synaptic connection. And you see that this takes place to, the object is to transfer neurotransmitters, and the neurotransmitters produce nervous impulses that transfer from one cell to another or to a muscle. And as you see, they're also encapsulated inside vesicles, okay? These are the molecules inside the vesicle of the neurotransmitter. And then there is a very, very small gap to the other side of the transmission, the other cell where they're making contact. And these neurotransmitter molecules are expelled when the vesicle, say, breaks, and then they reach receptors on the other side. And that's how the electrical impulses are transmitted. And here let me show a little bit below. No, I'm sorry, but here. Okay. Well, this picture shows several different types of neurons and also companion cells called glial cells, which in the brain are as numerous or more than the neurons themselves. So you have usual neurons, astrocytes, and oligodendrocytes, which are colored by different colors here. The blues are the neurons. Here you see a cell body. The astrocytes are in red, and the oligodendrocytes in green. Okay. Now, I think this is the last lecture part, is a paper which we published in 2013, that's in PLS1, which showed precisely what I mentioned at the end here, why this whole method of determining sigma and kappa is important, because they are connected with the cell function. Okay? So here we have very different cell types. And the forces here are piconewtons, and the radii in nanometers. Here's sigma in units of 10 to the minus 5 Newton per meter, and kappa is in energy units, 10 to the minus 19 joules. And you can see the values of radius, sigma, and kappa for each cell type. So you have astrocytes. Here is the U87Mg that we used in our study originally. This is a CX neuron, and we probed different parts of the neuron: the cell body, a neurite, and a growth cone. Then it's a GE neuron, again probed along the cell body or the neurite and along the growth cone. And then that was very important. We did this with microglia. What are microglia? They are glial cells that work in the brain just like macrophage works outside. You know, macrophage are kind of sentinels that when there is something that's harmful to the cell, they phagocytose it. Okay, they just engulf it, and then they will convey it to a way to be destroyed. Okay, now they do this depending on the medium, and in fact, for macrophage plus this amount of LPS, you get a value of the bending modulus of 8. And when you increase this, then you're activating the macrophage. And you notice that although the surface tension doesn't matter, doesn't change practically, the bending modulus decreases by a factor of 4. And that's consistent with the fact that that makes much easier to engulf what you want, what the cell wants to remove. Okay? So this is a direct relationship between the elastic constants and the cell function. Now, the same thing exists with microglia, where the microglia are like macrophage in the brain with the same function. And again, if they're not activated, the surface tension between activated and inactivated is almost similar, but again, the surface tension, the bending modulus decreases here by a factor of 3 roughly, and again, this facilitates removing what you want from the brain. Also, you can see the connection with cell function, because the elastic constants sigma and kappa for neurons are both very small. And they are very similar to those which we also measured for blebs. Blebs are just cell protrusions. For instance, I showed you the cell in apoptosis, it was blebbing. The surface was producing very small kind of bubbles. And so these blebs are essentially pure membrane. They don't have the underlying cushion, which is very important, which supports the membrane, which is called the cortex. Okay, so have no cortex support. And so there is a type of, well, a kind of artificial cells that mimics the blebs, which is PMV, and you see that the elastic constants for neurons are very similar. Well, they are up here, but they are very similar to those for blebs. And now this is consistent with the fact that the structure of the actin in neurons was found in a paper in 2013 by Xu. And you see that instead of having a cortex, okay, a kind of a, smooth curves like a mattress for the membrane, the actin is around in these circles, and therefore they have much less, they're more similar to pure membrane. Also, astrocytes and glioblastoma cells are very dynamic and mobile, and you find that they have much larger sigma and kappa. And microglia, as I mentioned, like the macrophages, undergo a large drop in bending modulus upon activation, favoring the phagocytic role, so the cell function and the elasticity are strongly correlated. So that's why it was so important to measure the elastic constants for all these cells. Okay? And that is what I mentioned at the end here. And I already covered the part about pathogens. Well, tomorrow I will show results about the role of CMN in cancer. And I can anticipate that just like with the other pathogens, the CMNs play a very important role in the propagation of cancer. Cancer propagates using cell membrane nanotubes. Okay? So that is all for today. And well, I can answer questions briefly now or then in the study group. Any questions that people, yes, but again, so that I can hear the question, I need a microphone for you. And please speak loud and slowly.
Oh, actually, it's more of a curiosity. I'd like to know about the differences between the studies for cell membranes, etc., to single molecule studies, if they're more in the data analysis part or also in the experimental setup. What are the differences between the membrane study?
Well, you can do experiments not with cells, but with artificial vesicles, okay? And then, of course, this is not connected with the the reaction. But you can compare those with the actual cells. Is that related to your question?
It's more related to, for example, Carlos Bustamante's group does studies.
Please speak slowly.
Okay, sorry. For example, there are studies where they study the unfolding of protein domains using optical tweezers. I'd like to know what is the difference between the data analysis, for example, or if there are differences in the experimental setup. If I want to study, for example, a protein in single molecule level to these membrane studies.
Oh, okay. Well, I mentioned yesterday, among the very, very important applications of optical tweezers, that you can use them to study single molecule interactions with cells. You can take, I gave some examples. You can, because you can go inside the cell, you can see how motor proteins move. You can measure forces and displacements following the path of these motor neurons, so directly molecule that's being followed. You can do this with and follow step by step how it's transcribed. All this is single cell biology, but we do not work with that. It would be very hard here in Brazil because you need very sophisticated techniques. Any others? Yes, there's a question there.
You mentioned in your first, thanks for the letter. So you mentioned in your presentation that the TNT could extend for like several cells in distance, right? So usually the adhesion of cells are short-range interactions. This TNT could create like long-range bonds between cells?
I'm not sure I understand the question. Perhaps if you speak more slowly, I will.
Okay. The cells adhesion is usually is a short-range force, right?
The what?
The adhesion between cells.
The cells what?
The adhesion between cells usually is a short-range interaction.
We measure these cells, but I lost the remaining. Can someone who hears better than I be
Prefere que eu fale em português?
Interpreter?
The adhesion between cells is usually a short-range interaction.
Oh, okay. That's a very good question.
Okay.
You can measure with optical tweezers the adhesion force between cells. Yes, that's an application that I did not discuss, but yes, it has been used for that purpose and can also measure how they attach to a substrate.
Okay.
Yes.
Okay, but my question is about the TNT, the tunnels.
How the TNT can contribute to long-range interactions between cells. Is it? How many?
Well, today I gave many examples of how they connect.
But these tunnels can attract the cells to make them come closer between each other and form a cluster of cells.
I'm not sure I understand.
Oh.
These experiments were done in vitro, but I mentioned that the same thing happens in vivo. Okay?
Oh,
so
okay.
Has this to do with your question?
Not exactly. Later, I talk with you. Okay?
Well, maybe in the study section, we can have a longer discussion, and maybe you come closer to me, and I understand what you're saying better.
Okay, okay. Sorry.
Questions?
Okay, so I forgot what time the study group is. Is it immediately now or? No, I think there's a coffee break, right?
Yes.
Is that correct? I think so.
Yes, that's right.
Okay, so we have a coffee break, and thank you for your attention.